Hello, everyone, and welcome to today's webinar, HPV-based cervical screening, from implementation to future possibilities. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Abbott Molecular. To learn more about our sponsor, please visit www.abbott.com. Now let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. Abbott will review the questions with Dr. Hawks and send the answers to the questions via email for the duration that the event is available on demand through LabRoots. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speaker, Dr. David Hawks, the Director of Molecular Biology and Biochemistry at the Victorian Cytology Service. For the complete biography on our speaker, please visit the Biography tab at the top of your screen. Dr. Hawks, you may now begin your presentation.
Hi, so today I'm going to be talking about HPV-based cervical screening from implementation to future possibilities. My name's David Hawks. I'm the Director of Molecular Biology and Biochemistry at VCS Pathology in Melbourne, Australia. I have my disclosures. We do a lot of research, so we've worked with most of the major manufacturers who've donated test kits, but neither VCS Pathology nor its staff have received any grants, honorarium, other financial in-kind gain from HPV and assay manufacturers. We've also been lucky enough to get a National Health and Medical Research Council grant from Australia over 5 years to be a centre for research excellence in cervical cancer control. So I guess the first thing to start off with is there's over 300 HPV tests available. I was recently at a conference in China where apparently there was over 200 tests available in China alone. And which ones are going to be suitable for use in a screening program is a topic I'm going to cover. Also, what's an appropriate quality framework for these tests? So we all know that a test is only as good as the staff that run it, but how can you make sure that your staff are actually following all the protocols and that the result is actually clinically valid? Another area that's sort of come up with HPV testing that wasn't previously available with cytology-based cervical screening programs is self-collection. So at the moment, it's used primarily as an alternative for primary screening, particularly with under-screened populations, but this is something that could actually grow, particularly in low and middle income countries. And the final topic I'm going to be talking about is HPV extended genotyping. And I guess this kind of comes down to whether more evidence is in fact better for a clinical purpose. So just to give you a little bit of background of the Australian experience, we have primary HPV nucleic acid-based testing for cervical cancer screening. We began on the 1st of December 2017. And we use a HPV-based primary screening with a reflex liquid-based cytology on any positive samples. As a result of that, we've moved from 2-year cervical cytology-based screening to 5-year HPV-based screening. And essentially the document that sort of drives what we do in the lab is the requirements for laboratories reporting cervical screening tests. Now, the first edition was released in June 2017, with the program beginning on the 1st of December 2017. And 17 months later, we've already produced a second edition, which I guess is a bit of a heads up to tell you that things change and they change pretty rapidly. And there's a lot of things in a national program we didn't predict that we've had to be reactive to. But we kind of went into this program knowing that this was going to be the case. We wouldn't be able to have a fully formed, perfect document outlining what we were going to be doing from day one. We're always going to have to be a bit flexible. So we've already got the second edition of this document. I sit on both this committee and also the original committee, and at the moment we're looking to sort of finalise that second edition, and we'll be actually go straight into working on the third edition. So this is just a general idea of the cervical screening pathway in Australia. So what I'll be dealing with most is in this blue box, which is essentially there's 4 possible outcomes to a HPV test. There's a HPV not detected, there's a HPV non-16, non-18 detected, and there's a HPV 16-18 detected, and then finally there's an unsatisfactory HPV test. I'm sure those of you who have been in the HPV or the cervical screening world over the last decade will have seen this figure, and this gives the cumulative incidence of SIN3, so cervical interepithelial neoplasia grade 3 or above, over 10 years, with the top line being the HPV 16 positive, the next line being the 18 positive. So these two in the Australian program, if a patient has a positive for either 16 or 18, they're considered at higher risk. Then we also have in the yellow, the non-16, 18, high-risk HPV positives. So there's still an elevated risk over HPV negative, but you can clearly see there's quite a large difference between the predictive value of a 16-18 and another. That other in this study was another 11 HPV types. In the Australian program, most other programs have sort of moved to 12 HPV types with the addition of HPV 66 and 68 on top of the WHO accredited 12 oncogenic types. So this is just a bit of a background into what we decided in Australia was going to be the requirements for a HPV assay. There's a number of other aspects that are particular to Australia that has to be registered with our Therapeutic Goods Administration, and these will change from country to country. But what I'm going to focus on is essentially the quality framework for choosing an assay. So it must satisfy an adapted Meyer criteria, which is essentially a test of sensitivity, specificity and reproducibility against a reference assay. Now, the reference assay is the hybrid capture 2 or the GP5+6+. We've now had one of the assays approved with the 4800 as a reference assay, and currently the Meyer criteria is being revisited by an international consortium. So that's why we're calling it an adapted Meyer criteria rather than in a strict Meyer criteria. It must be validated for primary population-based screening. The assay must contain a control to monitor inhibition and or assay failure. Now, this was a very controversial issue in Australia prior to the introduction of these requirements, and then it's become a non-issue. So for some assays, they test for a cellular protein, so beta globin is probably the most common, and that's used to measure inhibition traditionally by the assays, but it is also by default a measure of cellularity, which is the next point. So this beta globin can be used for both inhibition and cellularity. Some assays have different controls for both, but one single cellularity that shows amplification of cellular genes is more than adequate. And finally, there's the self-collection specimens must be tested using a DNA PCR-based test. The evidence for this was based on the 2014 meta-analysis by Mark Arbin, and since then the 2018 review of this has actually just strengthened that there's no difference in sensitivity between a PCR-based self-collected and a PCR-based clinician-collected assay. So according to the Australian program, these are the 7 assays that are currently able to be used in the program. Now, because it's a quality framework rather than selecting which assays and listing them, this can change, and this will certainly evolve over time. There's another number of assays that I suspect will be added to this list in the next probably 6 to 9 months. So the COBAS 4800, the Abbott Real-Time, the Hologic Aptamer, the BDL Clarity, the C-Gene, the Cepheid, and the Roche COBAS 6800. Now, when you look through these assays, and this is a table I always include because it's what I probably get asked the most, is we want just a single table that tells us what we can and can't do. And so this meets the the impact criteria, screening, cellularity. Now you notice there's an asterisk next to the hologic aptamer. That's because they have an IVD-based cellular control looking at mRNA expression of a human cell, and that's not validated by the manufacturer, and so it depends on what the local requirements are onto how adaptable that can be used. But it's also done as a separate sample, which means that the hologic aptamer on the Panther has a reduced throughput down to 130 in an 8-hour day. It's also not a PCR-based assay, so that can't be used for self-collection. All the others pretty much tick every box. And as you can see, there's a variation in the 8-hour workflow from the sort of the 288 of the Cobas 4800 and the Abbott real-time through to sort of the Cepheid Expert and the Cobas 6800. Now, the Cepheid Expert is a modular system, so you can have a single modular system which can do 7 in 8 hours, or you can have the Infinity 80, which has 80 modules, which can theoretically do 560 in 8 hours. Assays like the Roche Cobas 6800 can actually put on quite a lot of samples overnight. So just be aware the throughput in 8 hours is not necessarily that representative of the capacity of these machines in a real lab experience. But it is sort of the 8 hour is used as the metric for showing the volumes that these assays can actually turn through. So the other question I often get asked at international conferences, what are people using? So 2 large private pathology networks in Australia using a combination of the 2 Roche 4800 and 6800 systems. That's because in Australia we have long distances between all of our capital cities where most of the work is done, and different labs have different needs. So we have Sydney, which is a city over 5 million, whereas we have sort of smaller cities of only a couple of 100,000 where a 6800 wouldn't be necessary. Third private pathology system uses the Abbott M2000 system. The majority of public laboratories use a combination of the Roche 4800 and 6800. We've got 1 laboratory using the hologic aptamer with the IVD cellularity control, and 2 laboratories using the CG and Aniplex 2 system. So just to give you an idea of what BCS does, because we are sort of a pseudo screening reference laboratory, we are not a government laboratory, but we're also not a private laboratory. We are using both a 6800 and a 4800. We've got an Abbott M2000, a BD on Clarity, and a CG and Aniplex 2. These 5 instruments are used for our normal screening work. We also have an Abbott Expert. We have a point of care, but we also have a Infinity 80. We don't tend to use them because in a high volume laboratory, the cost per test is dearer, and it's not a sort of a financially viable option. But as a point of care device, it's certainly got some very big advantages. We've got an Abbott Alinity M coming in the next month, and then when the assay is registered in Q3 2019, we'll be using that for primary screening once we've got all the Maya criteria and the other quality framework items ticked off. There's a small Australian device called the Diagnostic, and we've also got the foundational HPV typing assay, the Roche Linear Array, but that's being discontinued next year. So as a result of having all of these tests and all these different targets, we're able to actually look into what these results mean into a wider context, particularly with screening. Now, we've already published 1 paper of our first sort of 116,000 screening tests, stratified by age. So our screening program runs from 25 to 74, and you can have a look. The top 2 tables are HPV 16 and 18, and the bottom table on the left is any HPV, and on the right is the non-16, 18 HPV. So what you can see here is that there's definitely a factor of age. So the younger women who are sort of, have started having different sexual partners, and then generally it settles down over time. So by 40, there's been a steep decline from 25 to 40 of the rate of infections. But what we can see is with Australia having such a high vaccination rate, over 80% for women, there is we don't see this peak in the HPV-16 and 18. And we expect as time goes on, this the 16 and 18 and the older women will actually start to drop away as the vaccinated cohort ages. The other thing we really want to make clear is that different assays give different results. So this is a paper published 6 years ago by Jack Cusack in British Journal of Cancer, and it shows that we've got the BD, we've got the Roche, we've got the hybrid capture, the Abbott, and the Aptham are all in there, and you look at the discordant results between the different tests, and you look at still looking about 0.8 on average between all these different assays. Now, if I was to see this on a chlamydia test, I'd probably be quite worried because chlamydia has an analyte control. Essentially, we want to detect any true positive in a chlamydia test. However, HPV is a risk stratification test rather than a diagnostic test. We don't want to detect every single positive in every single case because although a positive test does lead to investigation, not necessarily treatment, you're more likely to over-treat in particularly younger women because obviously that's where the higher rate of infections are. And it means biopsies or leaps or letzes on those women will most likely have an effect on their ability to carry a child to term and other sort of issues. So we only want to detect the HPVs that are associated with disease. And probably the most elegant way of showing this was a number of studies from Jesper Bond and Rebolgi, which show that when you look at the concordance of these four assays, which is a hybrid capture, Clark, Cobas, and Aptima, you've got 100 samples which have a less than SIN1, which is what we'd probably be looking at as normal cytology. Only 34% were positive for four assays. However, once you go to SIN2 or above, histologically confirmed, suddenly you're looking at 84%. And that's what we're looking at. And with this 84%, none of the positives are positive on just one test. So most of them are positive on 3. So it just shows that the tests really come together when you're looking at a HPV result associated with disease. But yeah, there absolutely will be some wobble when you're looking at sort of just infection that's not associated with disease. And we get a lot of questions about, so that's highlighting the SIN2. We get a lot of questions where a woman will have a HPV-16 infection. She'll go for a colposcopy. It could be a month or two later. They take a sample and she's negative for HPV. And what we often do is we can go back and look at the value, and often that initial HPV-16 is close to the cutoff within a couple of cycles of the cutoff of being defined as positive. That's how it works. That is just the nature of these types of assays. And it's not something that should be worried about, something that just needs education, particularly for the practitioners that are using it. And we've found that over the last 18 months, more and more practitioners are getting more and more comfortable with these sorts of results. Now, you've got a test, you're very happy with the test now, you've sort of got to work out how you make sure that that test behaves in the way that it should. So we know that sources of variability need controls to monitor the quality. So I'm just sort of run down a few of the general areas that we need to look at. And for anyone running a microbiology lab, this will be exactly as you have been doing for years and years and years. So we know that different instruments give different results. So we have a couple of 4800s. We know that they're slightly different. So the external QC controls need to be run on each machine that's being used. You can't just, if you had, say, 5 Abbott M2000s, you can't run the quality samples on just one and never test the other ones because you will get variations. We know that different reagents or different QC controls themselves can give different results. So we had a batch of a QC control where the manufacturer had put in the same amount of cells, but because the number of HPV copies varies per cell, we actually got a couple of, I think we got 3 or 4 cycles difference in our QC when we change batches. And so we're able to track that down and solve the issue and resolve it. But it just shows that the Q-keys consoles themselves can actually be a source of variation. We also know that different reagents can behave slightly differently. So what we do is we keep a record of all reagents producing a result, so that if we have an issue with any reagents, we can tell which results were actually produced at. And if we need to go back and look at whether the positivity has changed, we can actually track all that down. So that's basic data management. Pretty much Biology 101 is that all reagents and processes are possible sources of contamination. So on top of running positive and negative controls, we also run empty controls, but for us, that's generally a thin prep vial. And so we run those through our pre-analytics and our assays and everything else like that. And we'll run the same control for an entire week. And that monitors for any both HPV contamination, but also if there's any sort of cells, because these are amplification cells and they use amplification assays, and they use human cells. It's always been a source of concern that you'll get sort of skin cells or any other sort of cells giving false positives for the presence of beta globin. Now, we've been running those controls for 18 months, and we've never actually found cellular contamination of them, but we will continue to run them through the entire process pretty much every day. So the other thing to do is to monitor HPV positivity to detect changes. Sometimes you get variation. So if you get a particular younger cohort of women in, or you get a particular older cohort, you clearly will see differences in HPV. Now, different labs also have different population bases they serve and a variety of other parameters. So it's difficult to compare across labs, but certainly within your lab, you should have a bit of an idea of what your general positivity will be, and also the general breakdown of community. So in Australia, we consider anyone born on the 1st of July 1980 or afterwards part of what we call the vaccinated cohort. So they were people that were offered the HPV vaccine free as part of the Australian National Program. People born before that day, we consider the unvaccinated cohort. It doesn't mean that people couldn't have paid and got the vaccine, but just generally as a population base. And at the moment, that ties in very nicely with those women at 37, 38, which is about the point at which the HPV positivity is going down. But it's a date that we actually have to keep flexible, because obviously the vaccination of the women in that era only affects 16 and 18. And as we can see from the previous data, that's not where we're going to get most of our variation in positivity based on age. So as I said earlier, one of the things that changes with a HPV-based cervical screening program is the ability to actually use self-collection. To do cytology, you need to collect from particular areas from the cervix. You need to get a certain number of cells, and there's a lot of skill involved in this, and you need a practitioner to actually undertake it. With self-collection, it's much simpler. Now, in Australia, we have a number of regulations to make sure that self-collection is only offered to a subset of the population. So there must be over 30, must be more than 2 years overdue for screening, which at the moment means they're 4 years since their last PAP cytology, but within another year that will be start to be 7 years since their last HPV test. They must have refused practitioner collected, which is, a shorthand for saying a specular examination, and it must be collected within a clinical setting. So this isn't, in Australia, in other programs, there are mail-out self-collection kits. In Australia, it's all done at a clinical setting. So a practitioner will offer a woman a cervical collection, and she'll say no. They might offer them self-collection. She'll get the device and be able to take her own sample. So in terms of Australia, since 2018, under and never screened women, 2,000 have been able to access self-collection. The majority of those through BCS pathology, as we were the first accredited laboratory, and we're the only laboratory until relatively recently, in early 2019. And that's because we needed an IVD validation, because there were no PCR assays that have manufacturer-validated self-collection claims. Now, there is a variety of different self-collection devices available on the market. Some of them are very good from a quality point of view. Some of them, there's not a huge amount of evidence. We went with a Copen flock swab for a couple of reasons. Firstly, it's a relatively cheap device. It's under $1 in Australia. It's ubiquitous, so this is found in nearly every GP clinic in some way or another because it is used for, you can use it for nasopharyngeal. There's a whole variety of reasons that it's useful. So you don't need a particular self-collection specific device. And yeah, so the patient takes the dry swab and they put it back in the sleeve, and then it comes through to the pathology lab. Now, in terms of underscreening as a risk factor for cervical cancer, this is data from Victoria, and it's a report from 2015, so that's the most recent data we have, and it just sort of goes on to show that this is the group that is all underscreened. And as you can sort of see by the further highlights is of the invasive cancers, sort of the never screened and the lap screeners make up 70% of those. And it's almost exactly when you include invasive and microinvasive, again, it's 70%. Those that are sort of lapsed by less than 2.5 years, there's only one case. So it doesn't make that much of a difference. But yeah, 70% of women with cancer in Victoria are under or never screened. So your biggest risk factor for getting cervical cancer is not being screened. That's why self-collection is going to be a bit of a game changer in Australia. So in order to do a clinical validation of self-collection, we needed to know what was going to be a good assay. So we now know from the updated Harvard meta-analysis in 2018 that PCR-based assays are not less sensitive than clinician-collected. So it's 95% confidence interval of the largest for a different device was 0.87 to 1.04. It doesn't really matter whether you use a brush, a swab, or lavage. You're going to end up with a no less sensitive assay. There's marginally less specific, and that varies depending on the collection device. So when we're looking at a population that doesn't screen, I think there's less concern with the specificity, particularly when it's sort of very close, so it's 0.98 for a couple of devices, than it is with having something that's going to be less sensitive. So if you have something that, using an assay like the hybrid capture, you're actually looking at, they were statistically less sensitive, so they were only about 0.84, and that sort of was, again, statistically significant. So the PCR-based assays are the way to go. So the other thing to consider is the site of collection. So mail-outs for collection at home have a participant rate of about 20%. That's from the same meta-analysis. A study that we actually undertook with our underscreened population, offering it in a clinic, we had a participation rate of over 85%. Again, this is not a systematic review or a meta-analysis, it's just what we found. But yeah, we've also got anecdotal information from our actual national program that there is a very high uptake, which is why Australia is sort of sticking with the clinical offering rather than a mail out that's being used in other jurisdictions at the moment. So as I mentioned earlier, we had to get an IVD to use the self-collection in Australia. So the way that we did this was we undertook a self-collection study, which we called the SCOPE, and we did 303 samples. So this was to determine statistical non-inferiority. We'd had a previous study that had 42% positive, so that was what we based it on. We ended up getting over 50% positives in a colposcopy clinic, again, an enriched sample. So we met that power quite easily. We used self-collected versus a flock swab, stored for 7 days at room temperature prior to being resuspended 5 mL of preservicide, which is thin prep, by being swirled for 20 seconds. We wanted to keep this simple and easy. So we've done further stability where we've actually had that flock swab with a very low level of HPV at 50 degrees Celsius and 100% humidity for 2 weeks, taking samples out regularly and looking at the positivity, and they're perfectly fine. We're also testing it at -20 degrees, again, perfectly fine. So we sort of did 7 days originally, but we've actually done further stability studies on those. We then compared them with practitioner-collected thin prep using the collection device of the practitioner's choice, which is normally a broom or a brush. This is obviously given ethical approval, and this is sort of the data that we have on the Abbott M2000. So what you see is you've got some very good concordance. So if something's negative, it's generally negative on both. It's positive on both. And then we have a few samples that are self-collection positive or practitioner-collected negative, or self-collection negative, practitioner-collected positive. And what you can see is that there is definitely a trend that the self-collection is more sensitive. I'll explain why in a minute, but again, your 16 and 18, there's only 6. You know, you're still getting 26 positives for both of those, and it's, you know, HPV-18 is very similar. So there's not a huge degree of variation. And then when we look at all of those sort of self-collection, practitioner-collected as sort of a grouping rather than sort of just positive and negatives, there's, you know, same amount of 16s, one-off 18s, and very similar amount of other HPVs. Again, the self-collection is more sensitive, and the reason for this is that the self-collection was always taken first. This was a colposcopy clinic. Women who were going in for colposcopy may have had a biopsy or another treatment, and it was not considered either ethical or feasible to get them to take a self-collection afterwards. We know from other studies, such as this study by Pam Tollerman, where they did the same device, and sort of the vaginal specimens are on the left and the cervical specimens are on the other axis. We can see that in the red, this vaginal positive, cervical negatives were always much higher than the other way around. So it ended up being 54 were vaginal positive, cervical negative, and only 12 were the other way around. And that's because the first sample collected seems to be more sensitive, and whether it's a cervical or a vaginal, you seem to collect the HPV that's in the vaginal canal, and that's one of the drawbacks of our study, but it's also a well-known bias, and realistically we couldn't do too much about that. So the last topic I'm going to get to is about genotyping and the question about whether it's the future of screening. We know that longitudinal data is focused on either high risk, HPV positivity, or stratification of 16 and 18 and the other groups, as I mentioned before. So this is, again, one of those classic tables by Dillner, 2008, which shows the cumulative incidence of SIN3 comparing a HPV negative in red, cytology negative in blue, and a cyto negative, HPV negative in green. And there's 6 years data for this. The early study showed 10 years data. Which I'll just bring up again for ease of use. But the amount of data we have on further genotyping is still unclear. There's some small studies, but we certainly don't have the decade-long longitudinal data that we have. But what we do have is increasing commercial moderate to high volume automated assays with extended, and by extended I mean beyond 16 and 18 genotyping. So the BDON Clarity breaks down the 14 HPV types to 9 groups. The C-gene gives you all 14 individually. The Savide Expert takes your 14 types and breaks them into 5 groups. And your Linear EM takes your 14 types and breaks them into a slightly different 5 groups. So what we know is that these assays are going to develop over time, and we'll start to get more and more information. We know that some of the full genotyping assays have done 100,000 tests in some jurisdictions, but at the moment that's just sort of a snapshot of infection. And as I sort of showed earlier, we know that HPV is a risk stratification assay, not a diagnostic assay. So it's not quite as simple as just sort of showing concordance of infection with other infections. What we want to see is their predictive value going into the future. And as we don't really have lots of data on that at the moment, we've got to kind of look at it alternatively. So what I'm going to talk about is what is the evidence for the value of extended HPV genotyping in informing clinical decision making? Because I think that you can look at the research, you can have all sorts of arguments about different types, but at the end of the day, BCS is a pathology lab. what we're interested in is giving evidence-based clinical advice to our practitioners for them to assist in their decision-making. So I'm going to talk about Australian data just because there is a wealth of data, but this just sort of keeps it about as confined as we can. And we've also got a very strong screening program. So it sort of shows what happens, even in a good screening program. So there was a study called the ACT study, which was done, led by Julia Brotherton from BCS, and they took over 800 cancers over 10 years, finishing in 2015. So it's relatively recent data. And they found that 92.9% were HPV positive. Now, that means HPV wasn't detected in 7%. But again, these were histological specimens and they were slides and they were formalin fixed. So there is probably a degree of lack of sensitivity. That means that not all of these not detected would necessarily be negative. However, what we did find was that nearly 91% were positive for the 14 HPV types that we tested for. So these are cancers, not screening samples, but actual cancers. So between the 93% that were HPV positive and the 90.6% that were positive for the types we test with, there was only 2.3% difference. Now, a lot of people who are critical of HPV-based screening say that, well, most of the assays don't test for all of the HPV types, and that is absolutely true. But again, this is a screening test, not a diagnostic test, and we're detecting 90% of the types that cause cancers and missing 2.5%. Now, where we'll be in 10, 15, 20 years when we've had the full effect of things like the Gardasil 9 vaccine, this will have to be revisited, but at the moment, I think it's a fairly good predictor. Now, as you can see, the quadrivalent HPV and the Savarix types are covered by 16 and 18, which account for 70% of cancers. Then you've got the additional types in green covered by the quadrivalent. And as you can see, that covers overwhelmingly the cancers in the Australian context, as shown by this individual study. Then what I've done here is I've just broken it down slightly differently to what we're getting from our general genotyping. So most in Australia, the 16 and 18s go through one clinical pathway, the other HPV types go through a separate pathway. However, this is about 5,000 samples we've run with full genotyping, and as you can see that there's quite a difference between the prevalence of the HPV types that don't cause cancer and the ones that do cause cancer. So what you're looking at here is type 52 and type 58 are probably the most prevalent in this population. Very little HPV 18, a few other different types, but this sort of shows that the infection doesn't really meet up with the cancer too well. And I've actually added the figures from the graph a couple of slides ago. And it shows, obviously, we've got quite a lot of HPV-16, and that causes 52% of cancer. HPV-18, just under 20% of cancer. 31, 5.1%, 4.3%. When you look at 52, 0.2% of cancers were positive for that in our study. 0.6% were positive for 58. Even though we had nearly 8% positive for HPV-66, that was not being shown to be associated with any other cancers in our ACT study, and very few associated with HPV-68. So to just show this data again, so this is just the ratio of cancer to infection ratio. So there's 4 times as much cancer associated with HPV-16 than you'd expect from the actual infection rate. So about just over 10% infection, but over 50% of cancers. HPV-18 is even stronger with, just under 3% of infections being with HPV-18, but 20% associated, 20% of cancers. And you have a look, there's actually only one other HPV type that is enriched in cancer compared to the general population. That's HPV-45, which has always been acknowledged as probably the most likely to be sort of the next on the list after 16 and 18 due to sort of AIS and a few other different clinical reasons. Now, a number of the HPV assays, so like the Cepheid and the Aptam actually give a combined 1845, 1845 result for partial genotyping, and that's probably quite well founded. But at the moment, there doesn't seem to be a lot of reason to sort of base clinical judgments on individual genotyping. However, it's a small study out of Sweet Screen that actually shows that if somebody is persistent with a single type, within 7 years, all of these cases ended up with SIN2+. So as a screening test, I don't think there is enough evidence to suggest that we need to actually base clinical pathways on those results. But I think that certainly knowing if someone who's got sort of a persistent infection is persistent for the same type, there's definitely sort of emerging data showing that has value. Obviously, if you're looking at someone who's younger and having different sexual partners, the chances are if they've got a non-16, non-18, they might have different types, and that might help them avoid going for contiguous investigations. Or if you have someone who's in their 50s or 60s has had a long, stable partner, they're coming up with non-16, non-18, this might actually help that as well. So I think that just in summing up, trying to answer my own question of whether genotyping is the future of screening, currently there's not the depth of evidence to support risk stratification based on individual genotyping. That's at a screening level. There's increased amounts of data from national screening programs, so Australia, Portugal, and others, that the predictive value of individual genotyping will become clearer over time. So at the moment, we're sort of, when you're doing research studies where you have 40, 50, 60, 70,000 people, they're expensive and they take a long time, whereas with national programs in Australia, we've screened nearly probably over 2 million women, and we have a registry that monitors all of those sort of outcomes. So we'll be actually able to mine that data from programs. Portugal's moving towards a HPV-based screening program, and as I said, they've done a lot of full genotyping as part of one of their labs is working on that assay. The other thing is the effect of the national vaccination programs on the prevalence of 16 and 18 will likely play a role in the emerging value of extended genotyping. If you're only looking at 16 and 18, and 16 and 18 have actually dropped to negligible levels, then obviously the value of the other HPV types, particularly the Gardasil 9 included types, which we showed earlier, are associated with overwhelmingly the non-16, non-18 cancers. So that's 31, 33, 45, 52, and 58. These will likely be sort of the next group that will be extended genotyping playing a clinical role. So I'll just finish with my acknowledgements to thank the other staff at BCS Foundation, the Royal Women's Hospital Dysplasia Clinic in Melbourne, who allowed us to do the self-collection study, Cancer Council in New South Wales, who we've collaborated with on our number of our studies, and all the staff at the BCS Pathology Laboratory who actually do the work. Thank you.
Thank you, Dr. Hawks, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. Our first question is, based upon your experience, what has been the biggest challenges that your laboratory has faced upon the implementation of HPV-based primary screening program?
I think that there was a number of challenges in our program because it was very new and we were using a number of different assays. So learning how the quality control worked, being able to source that has become easier, but also there's little things that we never expected. So where in a busy day, we might do 1,600 tests, which means we have to get rid of 1,600 vials a month later, and every vial you have to uncap it and pour out the flammable liquids. And so actually getting the amount of waste we were processing became much, much larger than we probably expected. Generally, it actually went very smoothly. We used a variety of different devices. Our staff were well trained. They had competencies. We just sort of keeping records for all the different things that were happening in different pathways in the program were probably what we didn't expect. you'd be surprised at what doctors will end up ordering. But yeah, it was, from a lab point of view, there wasn't actually too much that was problematic.
Great, thank you. What are some of the lessons learned that you can share with the audience, given that there are still countries who are in the process or about to implement HPV-based cervical cancer primary screening programs?
Thanks. I think the one thing that we would absolutely recommend is communication is key. So the Australian program, we were probably not as good at communicating with our practitioners as we could have been, and definitely we've seen the level of education and the acceptance of the different program from our practitioners increase exponentially as we educated them. So I would sort of say start your education of your practitioners early, tell them what the test result will mean, even understanding what an unsatisfactory result means, so that the practitioner will go, well, what did I do wrong, and whether it was too much lubricant or not enough cells. All of these things are really what the practitioner wants to know. So education early and often.
Thank you. You had mentioned self-collection devices, which must be collected within a clinical setting. What type of education was required for the collection to be done correctly, and how involved was the laboratory in these efforts?
In Australia, the laboratory actually drives the self-collection. It is available on the program, but each laboratory has their own different way of doing it. We actually utilised a self-collection A4 one-sided how-to pamphlet, and everyone found it really, really easy, and it wasn't hard, and just showed a woman how to collect her own sample, what she needs to rotate it 3 times. We had practitioners going out and talking to groups of doctors, telling them, well, this is when it's appropriate and this is when it isn't, but a lot of those were specific to Australia and the conditions placed around self-collection. But yeah, it's been extremely easy, and it wasn't at all hard. And I mean, a lot of women, for a whole variety of reasons, have already self-collected for group 3 streptococcus, for in pregnancy, or chlamydia or gono, or any of those sorts of things. So it was actually probably the easiest part of the whole program.
Great, thank you. Your next question is, do you know what percentage of the population performs self-collection for cervical screening, and do you expect this number to increase?
So in Australia, as I said, we've had about 2,000 women out of about 2 million women who screen do self-collection. It's quite restricted, and there hasn't been widely available. We know that in Australia or in Victoria, about 87% of women have screened within 5 years. So that means at least 13% of women in Victoria, a state of just under 5 million, are eligible for screening. So we expect those numbers to go up. But when you look at programs like Malaysia is introducing a national HPV-based screening program, and it's going to be based on self-collection. So it will be all self-collection. So several million women doing self-collection. So I think that how many women are involved will depend on the individual country and how accessible it is. We certainly know that women much prefer self-collection. We're restricted into who we can test. So if a woman self-collects, we are not actually allowed to test it according to our accreditation unless she meets the criteria. Otherwise, I think we would have been doing at least 10 times as much self-collection.
Thank you. When the laboratory receives these self-collection devices to test for HPV, are there any additional steps that the lab needs to do prior to running the test?
So I think the thing is, it's very similar to getting any swab. So we get our swabs in, we put them in 5 mils of preserve site, we swirl it for 20 seconds, we take the swab out, then we can put that sample in the machine to go. So it's actually very straightforward. There's no pre-treatment, there's no any other different things we have to do.
All right, your next question is, what is your opinion on reporting out all the high-risk HPV genotypes, and do you believe that there is value in doing so?
I guess, I've spoken about this, and as I also showed, we've got a number of assays that we use routinely for screening that have different levels of genotyping. In Australia, the clinical outcomes are based on a 16 positive, an 18 positive, or another positive, and that's what we report. We do have full genotyping assays, and if we get a special request from a doctor that wants the individual genotyping, we'll put it as a note on the bottom of the report, but otherwise we just give the HPV result as it goes in with the clinical guidelines. We have a National Cervical Screening Register. We actually give them all of our results, so every individual type can be traced back retroactively if they need to, but at the moment, that's what we do. Other labs actually give every type, but we find that just leads to more confusion.
What is your opinion on the future of HPV beyond cervical cancer screening?
I think that there's a growing body of work with HPV associated with anal cancers, and certainly otopharyngeal cancers are becoming more and more interesting. There's certainly HPV associated with penile cancers, but it is generally at a fairly low rate. I think that as we get a really strong base and high volumes of HPV testing for cervical screening, the cost of the test will go down. It will allow these other cancers which are not quite as strongly linked to HPV as cervical cancer to be able to piggyback on those technologies. And yeah, I think screening for otopharyngeal cancer will be something that will occur maybe in sort of selective populations initially, but overall it will probably be something that will become more common in the next 5 to 10 years.
Thank you. What is your opinion about home collection for HPV testing, and what challenges or opportunities do you believe this will bring to the laboratory and for primary screening testing?
So I think home collection of HPV testing is great. There's a lot of studies, as I spoke about, that have used it. It's very well accepted, but the response rates are low. So it's about 20%. We've done a study here at BCS where we sent out several thousand of essentially a type of block swab, and we got 1,600 back, and that was great, but it's not quite as good as in a clinic. But from a lab point of view, it's the same device. It makes no difference whether we get it in from a clinic or whether we get it in from home. The only thing that we'd have to be aware of is that if someone collects it at home and they don't send it in immediately, there's just an increased risk of there being beyond the claims of validation. So if it doesn't get sent in for 3 or 4 weeks, we'd probably have difficulty testing it because we don't know of the stability of that sample.
Thank you. All right, your next question is, how big of an issue are lubricants when testing HPV, since there are various different types of lubricants out in the market? And are there certain lubricants that introduce more issues than others?
So I guess we've been quite lucky. Just before we moved to HPV-based screening, we had a short period in Australia where we moved to liquid-based cytology. And I know a number of other countries use liquid-based cytology routinely. Generally, we don't have a problem with lubricants because if you have a problem with lubricants, they'll impact the liquid-based cytology sample before they'll actually affect the HPV, just because the LBCs, the filters, and the other methods of actually processing the slide are more sensitive to it. We haven't had any particular lubricants that have been problematic. What we have done is we did a bit of study of unsatisfactory results a couple of years ago. What we actually found was overwhelmingly, I think there was 38 invalid results out of 22,000. Only 1 or 2 of them had sufficient cells and were inhibited due to the lubricant. All the others had insufficient cells. And so that's what we see as much more of an issue than lubricant per se. I know in a number of countries they don't actually even use lubricant. So I think that our practitioners recommend a 5-cent piece on the end of the speculum at most. But yeah, it's in terms of the different lubricants, it's not been an issue to a point where we've actually had to look into it. I know that for the liquid-based cytology, I think it's lubricant with carbon is in it, but it's not something we sort of see as much of an issue with the HPV assays.
Thank you, Dr. Hawks. Do you have any final comments for our audience?
No, I think that HPV is an amazing way to actually level the playing field for women that can actually be involved in cervical screening that was never possible with a cytology-based program. And yeah, it's sort of learn from people who've introduced it in other countries. There's a lot of things you'll never be sure of. There's a lot of good assays, but make sure that you have an assay that has a good quality framework around it. Thank you.
Thank you again, Dr. Hawks, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Abbott Molecular, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.